balance. In this session, I will be going to discuss about the introduction to acid-base balance, the first line control of the buffer system, the second line control, that is the lungs, and how the kidney is regulating the acid-base balance, that is the last line control. Then I will be explaining about the reabsorption of bicarbonate ions and how the new bicarbonate ions are generated, and a concept about limiting pH and anion gap. So in our body, the carbon dioxide, it reacts with water to form carbonic acid. In the presence of the enzyme carbonic anhydrase. So this reaction is taking place in our body, and the carbonic acid, it becomes H plus and bicarbonate. Here this H plus is the acid and this bicarbonate is the base. So regulation of these we are going to see. So in our body, when this level of H plus is increased, that means our body is going into acidic state. The bicarbonate which is present, it combines with the H plus and it form a buffer. It combines with a base, that is a bicarbonate. So this action is the buffering action, and this can be considered as a first line of control. It can be considered as a first line control. Why? Because this is acting within seconds. Because when the H plus is rising, suddenly it gets combined with the bicarbonate. So it is happening within seconds. So the next line of defense, the second line of control, it is by the lungs, because the lungs can increase or decrease the carbon dioxide level in our blood by altering the respiration, and so it take within minutes. So that is why it is called as second line of defense. I'll come into detail. As this CO2 is acidic, when there is hyperventilation, there is CO2 washout. So CO2 is acidic. So that means the blood will go into alkalosis. That is how it is maintaining. I will come into detail. The third level, the third line of control, it is by the kidneys. And how means the kidneys can excrete and reabsorb these bicarbonate ions. This capability help in acid base control by the kidneys. So the kidneys are having the capabilities to excrete. It can excrete H plus ion it can excrete the bicarbonate ion, and it can even reabsorb the bicarbonate ion. It can generate new bicarbonate ion. So by this mechanism, the kidney play a vital role in controlling the acid-base balance. And this is a last line of control for acid-base balance, and it take days to manifest. It take days to perform this action. So why our body is very conscious about the acid-base balance? It is because the pH. The normal body pH is 7.35 to 7.45. So if there is a increase in pH greater than 7.8, or if there is a decrease in pH that is less than 6.9, death is inevitable. It will lead to death. So why this pH changes? It is not good for our body. So one factor is the protein structure. So the change in pH can change the normal structure of our protein, and it can also lead to protein denaturation. Protein denaturation. And in our body, many enzymes are proteins. So it will lead to the denaturation of the enzymes. In this way, the pH changes in our bodies are sensitive to change in pH. So the second one, it will affect our central nervous system, especially the neurons. It will affect the function of the neurons. So during acidosis, for any reason, when your body is going into the acidotic stage, the central nervous system, it becomes less excitable. It becomes less excitable. And for any reasons, when our body is going for alkalosis stage, our central nervous system, it becomes more excitable. So the bicarbonate buffer system. So the carbon dioxide, it combines with water to form carbonic acid. In the presence of carbonic anhydrase, and it forms the H plus and bicarbonate ion. So this bicarbonate buffer, it is the most important extracellular buffer. It is the most important extracellular buffer. And when we are taking overall, this bicarbonate buffer, it is the most important one. It is because here the CO2 and this H plus, these two, the CO2 and the H plus, it can be tightly regulated. and with the help of the lungs and the kidneys. So the second buffer system is the protein buffer. So this protein buffer, they are most plentiful as intracellular buffer. Intracellular buffer. For example, when you are taking the RBC, the RBC contains hemoglobin, and this hemoglobin, it acts as buffer. So why this protein it is acting as buffer? It is because this protein has an amino acid histidine, and this histidine is the one which is playing a vital role in performing as a buffer. So the third buffer is about the phosphate buffer. So this phosphate buffer, it acts as, it is also an intracellular buffer. But the most numerous buffer, intracellular buffer is the protein buffer. And this phosphate buffer, it is very important as urinary buffer. It is most important of its buffering action in urine. So the fourth buffer is the ammonia, which is acting as a buffer. So in the urine, the ammonia act as a buffer, and it is also involved in the urinary as a urinary buffer. So these four buffer system discussed, they are important as a first line of control, and it takes seconds. So the second line of control, that is the lungs. How it is controlling the pH? So in lungs, no, the change in pH, it is not corrected by 100%. Only 75% correction can be done for pH changes, and it is mainly through the carbon dioxide levels. So we have two possibilities here. One is to increase the carbon dioxide level, and the other is to decrease the carbon dioxide level. So when you are decreasing the carbon dioxide level, that is, we can say that there is CO2 washout, and this is achieved by when you are hyperventilation. When you are hyperventilating, there is CO2 washout, and you can increase the CO2 level. You can build up the CO2 by hypoventilation. When you decrease your ventilation. So as I said earlier, that the CO2, it's like an acid. So if you are hyperventilating, there will be CO2 washout. That is, it is like the acid is removing from your body. So there will be alkalosis. So now explaining about the last line of defense, that is the kidney. So in the kidneys, as I have mentioned earlier, it can generate new bicarbonate ion and it can reabsorb the bicarbonate ion and it can even excrete the H plus ion, especially when the body is going into acidosis, and it can excrete bicarbonate ion when the body is going into alkalosis. So the mechanism will be explained. So in the PCT, the reabsorption of bicarbonate ion, 80% of the bicarbonate ion reabsorption takes place in the PCT. So we can see how. So this is the PCT. Here the side of the cell which is facing the blood side, this is called as basolateral side, and the side which is facing the urine side, this is the apical side of the cell. So due to metabolism, one of the cell of the PCT have carbon dioxide which combines with water and it forms carbonic acid with the help of the enzyme carbonic anhydrase, isoform type 2. So this carbonic anhydrase, it will be splitted into H plus and the bicarbonate ion. So this H plus, it goes to the urine side, and in turn one sodium comes inside through a protein channel, and this protein channel called as sodium H plus exchanger, sodium H plus exchanger. So the H plus reaches here and the bicarbonate which is filtered through the urine will be coming in urine, and this H plus and this bicarbonate, it will combine and form carbonic acid, and with the help of the enzyme, that is carbonic anhydrase type 4, and this carbonic anhydrase, it will be splitted into carbon dioxide and water, and this carbon dioxide, it goes inside the cell and this process continues. So in this way, the bicarbonate which was present here, you can see how it is reabsorbed. So here you can see this bicarbonate, it will be moved into the blood and it is reabsorbed. So with this, next is the generation of new bicarbonate ion. So new bicarbonate ions are also generated. So this is a cell in the PCT, and this is the blood side, and the part of the cell facing the blood side is the basolateral, and the part facing the urine side is the apical. So in the same way, in the cells of the PCT, there is carbon dioxide which combines with water in the presence of the enzyme carbonic anhydrase to form carbonic acid, and this carbonic acid will be split into H plus and the bicarbonate ion. So this H plus, it goes to the urine side, and in combining this H plus, earlier it was combining with a bicarbonate ion. So instead of that, now it combines with the phosphate, which is coming in the filtered, which is coming through the urine. So by this, it will become the HPO4 2 minus, and this is excreted. So in this way, you can see that the new bicarbonate is generated and it goes to the blood side. So the second mechanism by which new bicarbonate ion are generated. So this is a cell in PCT, and here we have the blood, and the part of the cell facing is the basolateral, and this is the apical side. So here we have the urine. So in the PCT cell, due to the metabolism of the glutamine, it will result to form 2 bicarbonate ion and ammonia. So this ammonia, it will go into the urine, and in the urine it combines with the H plus, and it forms the ammonium ion. So here this ammonia, it is acting as a buffer, and this ammonium ion, it will be excreted in the urine. So here you can see this newly generated 2 bicarbonate ion, it is not used in the urine as a buffer, and it is moving into the blood. That is how the new bicarbonate are again generated here. So in the above two mechanism, in generation of new bicarbonate ion, what you have seen is the phosphate, you are using buffer as a phosphate or ammonia as a buffer. So this, while acting as buffer, it is forming HPO4 2 minus and ammonium ion, and so these are acids, and these two acids are excreted. These type of acids are called as titratable acid. we actually have a limiting pH, and that limiting pH is 4.5. So when the limiting pH is reached, then there will be no more acid secretion, that is H plus secretion. But in PCT, we can see that this limiting pH, it is not reached in PCT. It is because in PCT the H plus is not free. It is buffered either with ammonia, phosphate or bicarbonate. But so in the kidney, where this limiting pH is reached, so this limiting pH is reached at the collecting duct, and that is why in the collecting duct there is acidification of urine. There is acidification of urine. So in the collecting duct, for acid base balance, we have intercalated cell. Intercalated cell, that is the I cell. So we have two type of intercalated cells. So one is the type A intercalated cell, which is acid excreting. So the acid excreting, it is mainly seen during acidosis, because in our body during acidosis we have to excrete acid. And we have type B intercalated cell, which is base excreting, and it is seen mainly during alkalosis. So here you can see this is a type A intercalated cell, and this is the side facing the blood, that is a basolateral, and this is the apical side. So the function of type A intercalated cell is to excrete acids, that is acid must go out. In exchange for that, a K plus ion, it will come inside, and this is by a channel protein, that is H plus K plus exchanger. And at the same time you can see that a bicarbonate is going inside the blood. In exchange for that, a chloride is coming inside. So this is by a channel protein, and that channel protein is the bicarbonate chloride exchanger. So this mechanism, it is mainly happening when the body is going into acidosis. So when blood is having more acid, it is excreting more H plus into the urine, and it is reabsorbing more bicarbonate into the blood. In the type B intercalated cells, here you are having the cell, and this is facing to the blood. This is the basolateral side, and here which is facing towards the urine is the apical side. So this is a type B intercalated cell. It is really a mirror image of the type A intercalated cell. That means, so in this case, it is especially useful during alkalosis. That means during alkalosis, you have to excrete more bicarbonate. For in exchange, the chloride is taken into the cell by the channel protein, that is bicarbonate chloride exchanger. And in the basolateral side, the H plus is going into the blood, and the K plus is going inside the cell by the channel protein H plus K plus exchanger. So here you can see that during alkalosis, more bicarbonate is excreted and more H plus is taken into the blood. So you can see that we are having the type A intercalated cell and the type B intercalated cell, and it is interconverting. That is, during acidosis, this type A is predominating, and during alkalosis, this type B is predominating. So this interconversion, it is made possible by an important protein, that is hensin. So this protein is responsible for this action. So now what is this anion gap? So the anion gap is, we have unmeasured anion. That is, in the routine lab practices, we are not measuring certain anions such as the protein anions, the sulfate, the lactate, and the keto acids. So these anions are not measured in our routinely clinical practices. So it results in the formation of an anion gap, and this anion gap, it is expressed in milliequivalents per liter, and it is equal to the sodium minus the measured anion, that is the bicarbonate and chloride. So this is how we measure the anion gap. So the sodium, it is nearly 140 milliequivalents per liter, whereas the bicarbonate is 25 and the chloride in our body is 105 milliequivalents per liter. and this comes out to be 140 minus 130, that is equal to 10 milliequivalents per liter. So this is the anion gap. So the normal anion gap, it is 8 to 10 milliequivalents per liter. So why this anion gap is important? So this anion gap, so during metabolic acidosis, the level of H plus rises and the level of bicarbonate falls. So for maintaining the electrical neutrality, the anion must rise, and if that rising anion is unmeasured anion, you can see that the anion gap, it is getting increased, and so such condition is called as high anion gap metabolic acidosis. Due to metabolic acidosis, there is high anion gap, otherwise called as HAGMA. So what are the causes for this high anion gap metabolic acidosis? So one reason is the diabetic ketoacidosis. Here the keto acids. The second cause is the salicylate poisoning. And the third cause is lactic acidosis. So these are the cause for high anion gap metabolic acidosis. So during the metabolic acidosis, there is increase in H plus. So in this time, if the anion rising is the measured anion, if the measured anion is rising, for example the chloride, which is a measured one, if it is rising, then the anion gap, it will be normal. The anion gap will be normal, and this condition is called normal anion gap metabolic acidosis, that is NAGMA. So here you can see that as chloride is rising, we can call it as hyperchloremic metabolic acidosis, because as the level of chloride is rising, and this is seen in diarrhea, in renal tubular, in the renal tubular acidosis, and it is also seen in when hypoaldosteronism, that is aldosterone is decreased. So in this equation you can see that why when bicarbonate ion is decreasing, when the chloride level is increasing, there is normal anion gap. So by this I'm ending this session. So finally we have discussed about the first line of control acting as buffer system, the second line of control, that is the lungs, and the last line of control, that is the kidneys.